So yeah, thank you very much, Thomas, for the kind introduction. I'm going to be speaking today about some of the work that's taken place in my lab at the University of Strathclyde. This work is done by a fairly significant multidisciplinary team. I won't go through everyone's research because we would be here all day, but I've tried to highlight some of the key words in case there's something that's of interest to you. I'm around until about Wednesday. Please come and find me. I'm happy to chat about anyone's work that's here. But really, our work scales from single molecule right through to mesoscale imaging, and it's mesoscale imaging that I'm going to be talking about today. And the mesoscale imaging arises because we want to occasionally study objects that are larger than a normal light microscope can see without compromising on the amount of resolution detail. So this specimen here is an image, so it's showing an image of a section about 16 micron thick of mouse kidney, and it's been stained with 3 fluorophores, but only 2 are visible here, and apologies for it being in red-green. These are not my data. These are data from Kurt Thorne's lab when he was at UCSF, and he's now made the sensible jump to industry. But it shows very keenly some of the problems when you're imaging on scales that are multi-millimeters or even centimeters in size. So the first problem that you see potentially is these dark borders that surround each one of the individual image panes. Now, Kurt here has very wisely used stitching and tiling. Stitching and tiling is great because you can take an image with a fairly modest magnification objective lens with a quite high numerical aperture, then you move the specimen, take another image and so on, and then computationally stitch and tile them together to form one larger image. But these dark bands can cause problems, especially for quantitative analysis of data sets. These data also obscure one of the main problems in stitching and tiling, which is registration. So from where you sit at your level of display zoom, you can see that, yep, you've got the gross anatomical presentation of data here. But when you digitally zoom in, what you might find is that in 2 dimensions, the individual image panes don't quite register next to each other. And in 3 dimensions, it can be even worse because you've got an extra degree of freedom. So while the image looks quite nice and we can tell what it's meant to be, often when we're putting these through our computational algorithms, the computers are struggling to see what we can see as humans by eye. So, as well as using stitching and tiling, there are alternative approaches to imaging of large tissues. So this is an acute example because it's now a commercially available option, and it uses random access scanning, where, for example, in the intact living mouse brain, it's possible to subsample specific volumes, small volumes within the overall large tissue volume, do this at very high speed, jump around, measure stochastic dynamic processes, and you can buy one of these. You can buy one of these from Thor Labs. It has quite a high price tag, but it's nice because it means that this technology is now being rolled out into the community and people are really starting to use multi-photon and mesoscopic scale imaging. Our approach to this has been quite different because most of the technologies that are out there for mesoscale imaging are based on multi-photon, mainly 2-photon excitation of fluorescence. But what we recognised was that there was still a need for going back to single photon and thinking a bit more widely about the problem. And we approached it as optical physicists do from an engineering perspective. So we returned to what the light microscope objective lens can normally do for you. And these are the really resolution profiles of the objective lenses that you'll find here or back in your own labs, but not in specimen space. These are in image space. So what you'll find is that irrespective of whether you have high numerical aperture, high mag, or low numerical aperture, low mag, these really resolution profiles look almost identical. The reason for that is historical. It's that they're designed for the visual acuity of the human eye. Where going back in time, the human eye was the photodetector in light microscopy. So with a 10 times eyepiece, again, in image space, excuse me, the visual acuity of the human eye is about 7 micron. So until the advent of digital photodetectors, cameras, photomultipliers, etc., there was no point in having an objective lens that could see more than your eye could detect. Now, these objective lenses are great. So we use them day in, day out. They have, especially at this end, very high spatial resolution in 3 dimensions, albeit within a sub-microscopic tissue volume. Or if you want to look at a large object, that's great, but your resolution's probably not going to be on the subcellular scale. If you're in the cellular scale, you tend to be doing quite well. Excuse me. So we approached this problem by looking at the Rayleigh resolution profile of these objective lenses and seeing what we could do to improve it. So this is the Rayleigh resolution profile of the Mesolens shown to scale next to the classical objectives. We've designed it to have a low magnification and high numerical aperture. So what that means is we've got a large volume of capture and subcellular resolution throughout that large tissue volume. This diagram here on the left-hand side shows the light path of a normal light microscope where the specimen sits here. You've got your objective lens, tube lens, eyepiece, and rather ineffective human eye as a detector. This is the Mesolens path shown next to it for scale. So it's roughly the same length and width as a human arm. Okay. Now what that means is that we can't just ship you one to your own lab. You're as well to come and use ours, or we can talk about how we can get you one. But it's not possible to bolt it onto an existing microscope. So we have to design the rest of the microscope to sit around it. We design the prescription using Zemax like all other objective lens designers. And then what happens is we take our prescription to a factory in Wales and we batch select each one of the individual glass types. There's 10 different types of glass in the Mesolens prescription. And we test it for low fluorescence. So here what we have is 2 pieces of supposedly BK7. And we've put a UV laser pointer through them. And what you'll see is that they're both wildly fluorescent. This one in the orange, this one in the green. And we can use neither of these in the Mesolens because that fluorescence would kill our image. Excuse me. If you like numbers, this is what we can do. So we have a 6 by 6 millimeter field of view, and we've got a 3 millimeter working distance, thanks to that 4 times magnification. We've got subcellular resolution in 3 dimensions, and we've got multiple correction collars. That means that we can immerse the specimen in oil, water, or glycerol. We can form an image in air, but the resolution isn't fantastic. All of this comes at a cost of fairly significant data volumes. So for a single colour, single optical section image, it's roughly half a gigabyte. So add in another couple of colours, that gets to 1.5 gigabytes. Think about a confocal series. A terabyte for a fixed specimen starts to become something that we need to deal with on a routine basis. I mentioned at the bottom here that the time taken to acquire data varies, and it depends on the method that we're using for detection. So when we conceived of the Mesolens objective, we thought about exclusively 3-dimensional imaging, and as I'll go on to explain in a moment or two, that's not quite the case. If you're familiar with the confocal principle, we use it identically to the point scanning method. So we have our laser, we expand the beam, we put it into a pair of scanning galvo mirrors, we pass the laser light through the objective lens and the scan lens, hopefully some fluorescence results. We then collect the fluorescence back up through a beam splitter, through a confocal pinhole, and onto a photo multiplier. And this is a photo of what it looks like here. It's roughly about a cubic metre or so in footprint. It's not like having an electron microscope where you need to knock out a wall and put in a sprung floor. Our scanning mirrors were borrowed, we'll say, from military technology. So these are roughly the same size as my hand. They're about a car wing mirror size. I don't think you can get them anymore, but they are extremely useful for us because we need to overfill the back aperture of our gigantic objective lens to make confocal scanning possible. So confocal is slow. Confocal is slow for us because we have to scan so many pixels per unit volume. So even with a very short pixel dwell time, say a quarter of a microsecond, it can take us 1 week to scan an entire Mesolens image volume. And so we tend to work quite a lot with fixed and cleared samples, as I'll describe. Although we conceived of the lens being exclusively used for 3 dimensions, as soon as we started talking to people about it, we realised that actually camera imaging was going to be useful too. So we needed a camera. The problem was that we needed a camera that had over 250 megapixels that were the right size for us to perform Nyquist sampled imaging. So this is the solution that we have at the moment. This is a camera that's normally used by the automotive industry for analysis of defects in large parts such as doors and wings, and it's based on a sensor shift. But rather than shifting the sensor by a full sensor width, we use it to subsample. So we move a sensor in a 3x3 array, moving the pixel by 1/3 of a pixel width each time, and we move this in X and Y. So we take a 28 megapixel sensor, and through some maths that looks a little bit like deconvolution, we end up with a sensor that's just under 260 megapixels, and the pixels are about the right size. If we shrink the field of view down to about 4.5 millimeters, we're now Nyquist sampled. So although I mentioned 1 week for imaging of confocal fixed cleared specimens, the camera's a lot faster. Excuse me. So this takes us about 10 seconds. There's now a faster version available. It should do about 3 frames per second. So it might be possible to put us into the space where we can do calcium imaging, millisecond exposures. So that's maybe one to watch. So I'd like to give some examples about how we're using the Mesolens in our own lab and with others at the moment. So I'll start with some kind of previous work to demonstrate what it can do. So the specimen here, and apologies to anybody that's red-green colourblind, is epithelial cell prep. The blue is DAPI stained cell nuclei, the green are fluorescein phalloidin stained F actin, and the red is mitotracker red. So we know from some simple image segmentation we've got 1201 cells in this field of view, and any of the zooming that I show you is entirely computational. The Mesolens is not a zoom lens, which means that unlike other stereo zoom systems where if you vary the magnification, you also change the numerical aperture. In the Mesolens, we don't. It's entirely fixed. We've always got that subcellular resolution. All I'm doing is showing you that through computational means here. So if we zoom in, we can see, for example, these cell nuclei are clearly resolved. We can see the bundles of F actin as part of the cell cytoskeleton, and we can see the bundles of mitochondria too. Let's see if we can move on. There we go. We've also used this not just for imaging fixed and cleared tissue, but even in camera imaging with fluorescence, contrast, we can start to make real inroads into cell biology. So roughly half of my group are now cell biologists, most of whom are microbiologists. It's a happy accident. I'm delighted about this. And what we're often looking at is ultrastructure in mature colony biofilms. This is what we see on the left-hand side here. So when you look at an E. coli biofilm by eye, it looks smooth and dome-shaped. So we assume that it's just a big clump of cells and there's nothing exciting happening in there. But using the Mesolens with wide-field camera-based imaging, we were able to detect for the first time this intricate network of channels that exists in 3 dimensions throughout the E. coli biofilm. And that's what's shown here. If I press play in a movie, hopefully we'll be able to zoom in and you can see them a little more clearly. So we didn't know why they existed. We've done a whole load of work out there to try and understand exactly the function of these channels. We now know that they're responsible for shuttling nutrients into the biomass, and on the right-hand side here, what we have are data where the colonies have been grown in different environmental and nutrient conditions to understand exactly how the morphology changes. And the preprint that's hopefully coming out in about the next 2 weeks will show some fractal analysis of these channel patterns, which is validating our results. We've also been able to use the camera for mesoscale imaging of standard histopathological tissue sections. So using red, green, blue colour contrast, we can do colour imaging, no problem at all. The specimen that we're looking at here is a section through the snout of a mouse embryo at full term. And these circular objects here are the whisker follicles of the vibrissae. So if we now take a region of interest, zoom in just to that region, we can see the vibrissae in more detail. And hopefully you can see, if we've got adequate contrast and resolution, the individual chromatin granules present within the cell nuclei. And that's the resolution that we have across that entire field of view. Coming back to our 3-dimensional imaging though, because that's really where it all started. These are some of the first images that we took and subsequently published. So this is a 12.5-day-old mouse embryo. It's been fixed, stained and cleared. The stains here are acridine orange, shown as the cell nuclei. The blue is a neuronal variant of beta tubulin, so this is an antibody labelled specimen. The field of view here is 6 millimeters. We're only going to optically section through about 200 micron, but we can do the whole mouse embryo. Just takes us a little while. And hopefully, if I press play in this movie, you'll be able to see, for example, features such as the ribs disappear, the liver come into view, and the edge of the brain eventually we start to see. And that's only over about, 200 micron thick or so. So we do have exquisite optical sectioning. And in case there's any doubt out there about what the resolution is, we're going to zoom into just this region here, the eye. And I'll show you what the resolution is there. So we have individual cell nuclei clearly present. We can follow the edge of the lens of the eye, and if I press play again, hopefully, we can follow, for example, the retina here. This is the edge of it, this is the edge of the lens of the eye, and you'll see that the retina eventually collapses, and then we'll go on out of field of view here to show the optic nerve. We've developed methods for preparation of specimens specifically for imaging with the Mesolens, but also now with other technologies, including larger stitching and tiling light sheet imaging systems. They include fixing and clearing whole adult Drosophila. This is a female fly, but I don't really like these data because while it looks quite nice, you don't see anything. Let's go to the raw data. So this is just a screen grab of me having a little look through the raw data in Image J. So let's pan down to the end, or at the thorax end, in the abdomen. Yep. So we can now simulate focusing. These are we know it's a female fly because these are the ovaries here and here, these giant nuclei, and we can pan back up through the thorax. We've got rows of flight muscle here, here. These are sections of leg. We can pan through the cardia. So this is just cleared in BAB, but it's nuclear markers. We've used prapidium iodide here. We can follow through the brain, the edge of the lens of the eye. We've eventually focused down through the mouth parts. So we've now got a full anatomical model without any dissection of the natural arrangements of the organs as they appear in the adult intact imago. So previous speakers spoke a lot about brain. Our brain imaging tends to be, as you can imagine, fixed and cleared, but we are imaging a kind of slightly larger spatial scale. So this is an ultra-thick section of mouse brain. The cyan contrast that you see here are DAPI-stained cell nuclei, but this is a mouse brain that's been challenged in an animal model of trypanosomiasis, where there are parasites in the mouse brain. The parasites here are constitutively expressing TD tomato as the reporter of the parasite fluorescence, and using the Mesolens we can then have a little look through these data sets. Now, the researcher posed the question, how do the parasites cross the blood-brain barrier? And their hypothesis was that they come up through the brain stem. So we were able to use the Mesolens, and these red blobs are the trypanosomes, to understand if there were any clustering phenomena taking place. Turns out there's not, so it's not coming up through the brainstem. But one advantage of the Mesolens, which we did not really anticipate here, was that it has the spatial resolution to stage the parasites and understand where they are in the replication in sexual phase. For example, these are short and stumpy, this one is elongated. So we can now start to better unpick exactly how the infection is spreading within the mouse brain. Because I keep talking about fixed and clear stuff, we are also imaging living specimens. So this is a Drosophila pupa. The hemocytes are weakly expressing green fluorescent protein. We've downsampled the field of view a little bit, and the researcher, who's based at University of Edinburgh, was interested in the flow of the hemocytes at this fairly late stage of the embryo development, of the larval development. And what you can see is this pulsatile behaviour where they're ejected from this bottom left-hand side here, collected, and they circulate round as you would expect in the circulatory system. Now, the advantage here is that the green fluorescent protein in these hemocytes is so low, but the collection efficiency of the Mesolens is about 20 times higher than any objective lens, we can use this for still quite high contrast imaging. So the researcher was delighted, and therefore we were pleased. We're also using the Mesolens for studying of clinical samples. So roughly twice a week, one of my students will go to the local hospital and collect human palatine tonsils. And what we want to do is really understand whether the rate of tonsillectomies that's been performed, certainly in the UK, is warranted. It's one of the most contentious surgeries that takes place because it takes place in children. And that's never great. But it has one of the highest post-operative bleed rates. Roughly 1 in 5 patients is going to be readmitted within 2 weeks. And it costs them a lot of money. It doesn't help with their social and educational development. So in the UK at least, it's certainly highly contested. So what we do is we get these tonsils, we stain them so they're still fresh at least, and we can use a combination of reflection mesoscopy, which you see here in green, and then thanks to fluorescence labelling, where we can label up, for example, the gram-positive bacteria, we can look at the bioburden. And roughly halfway through this movie, you'll see where the bacteria sit. So we're trying to understand exactly how effective the antimicrobial agents that are normally prescribed during pre-tonsillectomy are in preventing disease. And so this is ongoing work, but I wanted to show you at least some of the data that we've acquired so far. I mentioned that confocal is slow, and the way that people normally get around the slowness of confocal is by moving to a light sheet. The difficulty for us is that we need a light sheet that extends over about 5 millimeters or so, but that's only going to be a few microns thick to make adequate use of the axial sampling of the Mesolens. And technically and optically, that's a major challenge, because if you focus light to maybe a couple of micron, it's going to diverge very rapidly if it's a Gaussian beam. So we approached this problem using 2 methods. The first one was to say, right, let's live with a thick light sheet. We'll have a 30 micron thick light sheet. It won't vary in diameter very much across that field of view, and we could use it for fairly crude sampling. So we've done that. The second one was to be ridiculous, and we've developed a non-Gaussian light sheet, so it's propagation invariant. It's about 4.5 millimeters long. It extends from about 10 micron at the exterior to about 8 micron in the middle. So it's consistently flat. We do this using aspheric optics and putting them off axis. So this work is now published, but I'm happy to chat about it in more detail. But this accelerates our rate of image acquisition. So it takes that 1 week and takes it down to about 1 hour. And in doing so, we don't really compromise too much in spatial resolution. So this is another ultra thick section of mouse brain. It's cleared. And the fluorescence reporter here is for GFAP. So we're looking at an ancillary reporter of inflammation in the brain. And it's specifically targeting astrocytes, so we can look at the length of the projections of the astrocytes, perform some image analysis, and understand exactly the rate or scale of inflammation in this specimen. And we used the light sheet to do this, because otherwise it was just going to be far too slow. We've also used the Mesolens now in interferometric imaging. So the standard way of presenting the specimen to a normal microscope, which is not the Mesolens, we've developed new ways of doing that, is to sandwich the specimen between a slide and a cover slip. In our hands in interferometric imaging, what we do is we mount the specimen directly onto a mirror. And what we have then is an incoming illumination ray. It's reflected from the mirror. The specimen normally sits about here. It's retro-reflected back up, and in doing so we create planes of constructive and destructive interference in the microscope. The planes are separated by lambda over 2n, where n is the refractive index of the mountain or sample, and each plane is separated by less than 1/4 of a wavelength thick. So in doing this, we can build up a topographical map of the cell membrane or interior organelles. And we've done this at the micro scale, just to show you what red blood cells look like. They've got a kind of curvature of the membrane. This is a healthy red cell here, where you can see that as the contour lines come up, you're coming up in the basal membrane. This is not a happy red cell. You can see these projections. These are not visible when we don't use interferometric imaging. But we've extended this to the mesoscale, And we've also added in more wavelengths so that we can try and fill in the information gaps. And in doing so, we can build topographical maps of hundreds of cells simultaneously. We've got a couple of regions of interest here. If we go to this one here, you'll be able to see, for example, where there are dark regions. We believe these are focal adhesions because they are adjacent to where the mirror sits. But we can tell by looking at the colour lookup here, this is a red, green, blue colour merge. We can tell exactly the distance from the mirror itself. Because the previous speaker was talking about multiphoton, I realised that there might be an opportunity to speak about some completely unpublished work. I'll publish it eventually whenever we can. And it's trying to upscale the Mesolens for multiphoton mesoscopy. So some years ago, we published a method using wide field microscopy, where we took a beam of light from a Thai sapphire, focused into the back focal plane of the objective lens to create a stationary and parallel beam. We used that for excitation of fluorescence, and we were able to leave that sitting in the specimen, not scan it around, just constant wide field illumination. And by virtue of the fact that our camera exposure time is a lot longer than the pixel dwell time of a laser scanning microscope, you don't compromise in the amount of fluorescence that ultimately you are generating. And we can make reasonable contrast images and image at high speeds, so we can do 100 frames per second, for example, in the microscope. We've now extended this to the Mesolens. So our initial thought was to do it exactly the same way. Bring the ultra short pulse in through the objective lens and excite fluorescence using this parallel beam of light. That was never going to work. And the simple reason it was never going to work is because there's too much glass in the Mesolens and the pulse stretches prodigiously. So that was never going to really work for us at all. But as a proof of concept, what we've done is expand the Thai sapphire up, introduce it in a transmission path through the Mesolens itself, and use the Mesolens as the imaging objective to collect the fluorescence that's a consequence of the two-photon excitation. So we extend this beam to about 4.5 millimeters across the field of view, and then just use the epi-detection path of the Mesolens. And it works. So these are water fleas that are stained with eosin. So the field of view here is 4.4 by 3 millimeters, and we've got two-photon excitation of fluorescence. But if you know anything about water fleas, you'll notice that, well, they're quite big. I mean, these are maybe about 1 millimeter or so in diameter. They're also thick. And at the moment, we don't have optical section. However, it should be possible for us to adopt the method of Aron and Silberberg, who published temporal focusing about 20 years ago now, in the optical path. And it should be possible for us to then perform optical sectioning using the Mesolens, at least in transmission mode. And we have a gradient of 2, we've done all the usual checks, it is two-photon excitation of fluorescence, and that's great. Now, you should probably be asking yourselves at this point, why would you bother? What's the point in this? So we have high resolution, that's all great, but why not just do single photon excitation? So I'm going to press play in this movie, and what you'll see is nothing happening. That is because we have almost no photobleaching at all with this two-photon mesoscopy approach. I'll play it again, just in case you don't believe me. Over about 10 minutes or so, we lose less than 1% of our fluorescence intensity. So this is, bearing in mind that we're applying across that full field of view 4.5 watts of femtosecond pulse Thai sapphire light. Maybe we need to start thinking again about photophysics of ordinary dyes, because there's something interesting going on here. Okay, so I'm going to finish up by telling you what happens next. So we have this facility at Strathclyde. As I mentioned, it's not really easy for us to post you a lens, but we do have a facility. Send us your samples. We're really happy to look at them and send you data back. We collaborate widely. We've got all of these different modes. If we don't have the imaging mode that you want, we'd be really happy to talk to you about how we can develop it, because that's what keeps me in a job. The next thing that we're doing is we're creating a new Mesolens totally. So the images that I've shown you are nice. We've got 6 millimeter field of view, we've got a 3 millimeter working distance, but often what's happening is we're giving the biologists back their data, or the researcher back their data, and they're saying, this is nice, but I just want to see a bit more. Now, by more, what they normally mean is more of the sample. It's not really more resolution that they're seeking. So we have a new Mesolens. It is chromatically corrected across the entire visible spectrum, and into the near-infrared because we want to do multi-photon, also through the objective lens because we think we know how we're going to correct for material dispersion. It extends the volume of capture from 6 by 6 by 3 millimeters to 6 by 6 by 11. So we should be able to image the whole mouse brain with subcellular resolution, multi-immersion like the last one, and it should be compatible with all the modes. And we've been able to model because we know exactly what's in this objective lens. Unlike every other objective lens in our hands, we know what's in this one. So we can theoretically predict how the light's going to propagate through it and make sure that it's going to work. This is what it looks like. So at the moment, it's not really adequate because the eyepiece is roughly where your knee is. So we're going to have some big ceilings, but that's not impossible. This is one of our team here shown to scale next to it. The camera sits up here, Mesolens here, stage, yeah, and it's working. So this is good. Except there's a problem. So the problem is that it takes roughly a year and a half to make one of these things because we don't have the engineering capability at Strathclyde. So we farm this out to a third party and then all bets are off. We need to wait for them to give us the lens and then we turn it into a microscope. So we've turned our attention to how we might be able to improve this manufacture process. So I know that you've got a fab lab out here and I asked the question last night, are you able to print glass? And I don't think that you can, but that's what we're now doing. So we are now, and hopefully this one will come out in preprint, watch bioarchive in the next week or two, we are printing our own optical elements. Now, at the moment, these are small in diameter. They're about 20 millimeters or so. And I've brought a few here in case anyone wants to come and see them. So they perform almost as per a standard glass lens. So the ones that we print cost us 11 pence per lens. The ones that we buy from Thorlabs cost us close to €100. It takes us about an hour to make maybe 12, 15 lenses. So, but they compare really well. We've got good high optical throughput, over 90% for most of the wavelengths we want to use in light microscopy. We've got good control over the curvature, focal lengths, and they focus light to a diffraction limited spot, more or less. So watch this space. We've also been able to use them for imaging. At the moment, it's not mesoscale imaging, but that's where we want to go next. We think we can do better in terms of the resolution performance, but at the moment, it's on the micron scale. We think we can improve on this greatly. So with that, I'm going to finish up. I want to thank everyone at Strathclyde and all of our collaborators, not all of whom are mentioned here, for sending us samples and working with us. It's been a long slog to get to this point, but we're certainly not finished yet. I want to thank all of the funders for making this work possible because we can't do anything without money. And I want to finish by advertising yet another course. It won't be in the same scale as this one, a bit more basic perhaps, but it will take place in Scotland next summer. So if anyone wants to chat to me about that, I'd be very happy to do so, and I'm happy to finish also.
Fantastic talk, thanks. I might have missed it, but what is the working distance of your Mesolens? Because this screams for expansion microscopy, right? And we are interested in tissue blocks of around 1 millimeter, so if you expand them 4 to 10-fold, you need that type of working distances.
So, excuse me, so the working distance of the lens is 3 millimeters. And you're right about expansion microscopy. We're now working on it.
Okay. Thanks.
Thank you.
Can you tell us more about the processing that you use for the 3D printing optics that you showed in the end, at the very last slide? Like what kind of processing? Is it chemical? Is it mechanical? Is it both?
So this is resin printing. So we've tried with extrusion, doesn't work so well because you get the scattering of light. So we make a form in the resin print, and then the post-print, post-cure step, I should say, is spin coating with a slightly different resin, and that allows us to produce the smooth finish that's required to minimise the scatter of light.
So you spin coat on the printed elements?
Yes, so it's highly scattering without the spin coat. Once the spin coat goes on, it looks like a lens. And they're hard, you can drop them, you know, it's fine. They're as hard as any other lens.
Other questions?
Thanks a lot. I was pretty surprised or startled. For sure, there's something I don't understand about the very low bleaching rate you showed. So, okay, you mentioned that you are applying 4 watt, but of course it's a much larger field of view. So, basically, what I would like, if you could elaborate on that issue, because there are many things which just are speaking against intuition and things which we are used to seeing in that context.
So I agree with you. I had expected that the bleaching would behave probably non-linearly like the work of Patterson and Piston. Okay. What we found at the micro scale, forgetting about the Mesolens work that I've just presented, is that the bleaching also was a lot lower than we expected compared to single photons. So we've already published this 2016, so we can chat about that one later. I'm glad that you've noticed that it is 4.5 watts, but it's across 4.5 millimeters. So overall, the photon density is low. But I think this just is speaking to the fact that there is something going on. We don't know what it is. So yeah, maybe we should chat later, because we just don't know.
We have time for other questions. So I have another one regarding the very large light sheets that you were using. You said you were using aspherical and off-axis alignment. Can you also elaborate on this?
Yes, I can. So for the long, thin light sheet, we tried several methods, some of which had already been published, which we struggled to implement in our own lab. And we ended up with a combination of an off-axis Pupil lens and I think it was an on-axis cylindrical lens. So we modelled it in Zemax. We struggled to model it in Zemax. It didn't really do what we needed it to do, and it almost ended up being an empirical observation in the lab, which we then built upon, characterised, and it behaved quite well. We call it an Airy-like light sheet because it has the same optical path-ish as an Airy-like beam, where you have the first maximum and then some subsidiary minima. The difference here is that because it's over such a wide field of view, we don't really see the curvature that you normally see, this banana shape that you normally get in the Airy beam. So we don't really have that. That's why we don't call it an Airy beam per se. The other thing is that because we have such a small depth of field for the Mesolens, so it's 7 micron, we don't pick up the subsidiary beamlets in the axial direction. So we don't need to perform any post-acquisition deconvolution to remove those from the data set, because we only detect that first order, or 0th order, I should say. That simplifies things enormously, because then you can immediately use your data set rather than having to work with tens or hundreds of gigabytes to process it before then you can analyse it.
And it's not an issue with light sheet to have such a small depth of focus?
Sorry?
Usually in light sheet you want like a quite large depth of focus to like kind of get rid of the alignment issues, or?
Yeah, so we would love to have a big depth of focus, but or depth of field here, because that would simplify all of our setup. It's difficult to align, but once it's aligned, it works perfectly. So yeah, it's good.